Hello, Bruins. This is Mrs. Farouk, and this is part two of Darwin's ideas. And we're going to talk about evidence of evolution. What are some evidence that we support the theory of evolution? And we're going to look at fossils and structures, embryological and molecular evidence. Okay. So evidence of evolution is what we look at is the fossil record, and we could see that the fossil record shows the past living organisms that are no longer present, and there are some that are similar to present organisms. And fossil, so that's based on our observations. Now fossils, there are many different types that can give us clues about the past. Trace fossils show animal activity. Cast fossils are minerals that fill up the space left by the decaying organisms. We have molds. When organisms were buried, the sediment filled in, leaving an empty space. And then we have petrified, where the minerals replace the hard parts of an organism. And then there's amber or frozen, that a whole entire organism is trapped in that amber or ice. Now it's really difficult to have fossils records of invertebrates that don't have like bones, so there's a lot of gaps with the fossil record. Now if you look at life's natural history, you can see that there were a lot of successions and extinctions in the past compared to the present organisms. Now fossil record also show that the present organism has changed over time. So here, for example, Darwin noticed the extinct giant ground sloth was much bigger, say, compared to the modern sloth, which is much smaller. So you could say that he thought that there was a relationship also in terms of where he found the fossils and where the present organisms are found. Now most of the fossils are found in sedimentary rocks, which are most likely to contain those fossils because they slowly accumulate over time, and that compresses to form the sedimentary rocks. Other types of rock would just totally destroy whatever organism there is in there. So the fossil record gives us a relative idea. If you look at the different layers, so as you go deeper layers, you could look at the older organisms versus the more top layers are the more younger organisms. So this is something what we call relative dating. Now more accurate dating is the radiometric dating, where every atom, if you remember from the beginning of the year, have isotopes, and so basically every material decays, and it decays in a particular amount of time, and that's what we call half-life. So say for example an atom can have, say, 5,000 years. So after 5,000 years, what this represents in red is the parental material, and half of that decays. So now you have half parental and half what you call daughter material. Then another 5,000 years, another half of that parental material decays. And then another 5,000, another half of that. So you can figure out the difference between the parental material and the daughter material, and see the ratio to figure out how old is that fossil. Now we also looked at that organisms are isolated in continents as they drifted. So you can see similar organisms on the coast, say, of Africa and South America, but then over time, because they have drifted, you'll see differences, and some animals, as you can see, could not adapt, which led to extinction. Some did, so you see similarities and differences. Or those animals that were isolated in islands like Australia, we would see unique animals. So this is what Darwin observed in unique species in geographic isolations, like we saw in Galapagos, like the big tortoise or the blue-footed booby birds and the finches. So this is another thing we call BioGeography, where in similar environments you would find similar animals even though they have different ancestors. So for example, here we have the flying squirrel, and you have the sugar glider. They both look similar, but one is a marsupial and one is a mammal. I mean, they're both mammals, but they look similar because they have living in similar environments, yet they don't share that direct common ancestor. So this is an example of convergent evolution, where two different animals are starting to look the same in similar environments. You also see that, for example, in dolphins and whales versus sharks. They all got that streamlined body because they live in that similar environments, yet one is a mammal and the other one is a fish. So looking at biogeography, we'll look at fossils and seeing similarities and differences along the coast in different areas. Another evidence of evolution is we look at homology. Homology results because of common ancestry, meaning that there's evidence that they're common ancestor because your structures are similar, even though their function is different. So if you look at the human hand, the cat, the whale, the bat, they all have that similar structure. So they all have that humerus, radius and ulna, carpal, metacarpals, and phalanges. Yet they might be a little different, but the structures are all there. Now, opposite of homology is analogous. Analogous means they have the same function, but the structure is different. So if we look at here, like sharks and dolphins, they have similar shape, their skin, again, adapted for swimming, to live underwater, yet they have different structures. Another evidence is looking at vestigial structures, which are remnants of features that served important function in an organism's ancestor. So for example, why would a whale have hind feet, or a pelvis and femur even though it doesn't use it? Or why would a snake have a pelvis and a femur? Now, we also can look at embryology that reveals similar structures. So if you, here is a picture of different organisms. So in the beginning stages, they look very similar, and then they start differentiating in the later stages. So here's a chick embryo, and then compare that to a human embryo. They both have those pouches, pharyngeal pouches, and gills, and they both have that post-anal tail. And then most important evidence is the molecular evidence that there are similarities in molecular structures, the DNA, or you can look at the protein, looking at the different amino acid sequences. So as more closely related the animals are, the more they share, and the further they're apart, the more differences there are. So scientists also have studied over a longer period of time to see how animals change. So here's a classic experiment where they looked at guppies and predators. So here in one pool, they have this predator killifish that preys on small guppies. So they found in these pools that the guppies are much larger at sexual maturity. And then another pool, the predator preys on the large guppies. So these guppies mature sexually, and they're smaller. So when they transplant and move these guppies into another pool with killifish but no guppies prior to transplant, they saw that changes, and after 11 years they saw how the size and the age at maturity changed. So many studies have been taking place as places such as this and the Galapagos to see how the environment affects phenotypes and how over that particular time their phenotype changes. And that's what we mean by evolution, change over time. See, in your notes, if you could summarize, here we have bugs, and as you can see, there's variation amongst these bugs. How do these bugs change over time? And the environment here, the environmental pressure here is the pesticides. So when you are describing how the pesticide selects for which bugs survive, and at the end, how does that population change? So you look at using keywords as variation, natural selection, environment, adaptations. And we'll discuss this in class. Thank you for listening.